Yes, good morning to you all. Yes, we have started the revision of the chapter Fundamental Unit of Life. Yes, in the last day, in the Tuesday, we have studied about the fundamental unit of life, and we have studied about different types of cells that we have present in plant cells, animal cells, all those things. Okay. So we have studied about all those things that we have, and we have studied about plasma membrane, that means the cell membrane, then about the cell wall. After all that, we have studied about cytoplasm, and we get into the next section, nucleus. And after all, we get into at last, in I think in the when we are stopping, when we stop the class, we have studied about the cell organelles. Okay, studied about the cell organelles. So what we have to say about cell organelles? Okay. Okay, what we have to say about cell organelles? Yes, we have studied about something that we have to know more about, and we said that we already finished the chapter and we are revising now, so you know about something about the cell organelles, or the different types of cell organelles we have present. So I think it have been 1 week, or it had been 1 week that we finished our class of cell organelles, and we are repeating now. Okay, so I think you can refresh this, and we can write an exam for that after all that. So when we say about cell organelles, what we all have said, every cell has a membrane around it to keep its own content separate from the external environment. So when we are saying about that, we can say that large and complex cells. Okay, there will be large and complex cells, that including cells from multicellular organisms. Okay, including cells from multicellular organisms. So it's need of a what we say, variety types of chemical activities. Okay, variety type of chemical activities is taking place inside the cell. Okay, so it needed the complicated structure and function to the performance. Okay, for performance it need a complicated structure. So to keep these activities, or we have to say that there will be different type of structures will be present, or these cells use membrane bound till little structures. Okay, they will be used membrane bound little structure for, it means within themselves. Okay, within themselves, that is known as organelles. Okay, organelles, what that we said. So these little structures, one of the main feature when we are saying about a eukaryotic cell, when we are saying about a eukaryotic cell, we have said that we can easily distinguish eukaryotic cells and prokaryotic cells using the cell organelles. Okay, and it is very visible. The cell organelles are very visible using the electronscope microscope. Okay, electron microscope only. If we are getting a compound microscope or normal microscope, we can't see the cell organelles. If we are looking through a compound or electron microscope only, we can look into the cell organelles. Okay, so we have said about nucleus in the previous class, and we have want to say something, we already said out some examples of cell organelles, and that are endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastid, and vacuoles. Okay, in last class we have said about that. So we have talked about different things, that is endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastid, and vacuoles in cell organelles. Okay, so we are going to study about that. Okay, so we want to start about the endoplasmic reticulum. What is the specialty of endoplasmic reticulum? So about the endoplasmic reticulum, we can say that the endoplasmic reticulum is a large network of membrane bound tubes. Okay, it is a large network of membrane bound tubes and sheets. So what we said, that it looks like long tubules. Okay, it looks like long tubules or round or sheets look like, okay, round or oblong bags, okay. It looks like a vesicle, or it looks like a long tubules. So that's its the speciality. So the when we are looking an endoplasmic reticulum, we can say it is very similar to the plasma membrane that we have studied. Okay, the structure of endoplasmic reticulum is very similar to the plasma membrane. Okay, so that's all about that. And as we said about endoplasmic reticulum, there are two types of endoplasmic reticulum, that is RER and SER. RER and SER. RER means rough endoplasmic reticulum and SER means smooth endoplasmic reticulum. So RER looks like rough itself. Okay, that's why it's named. So RER looks rough under a microscope because its particles called ribosomes. Ribosomes will be attached to the surface of the rough endoplasmic reticulum. So it will be looks like rough. And the ribosomes which are present in all active cells. Okay, sites of protein manufacture. So it is as you know, the ribosomes are the main structure that is used for the protein manufacture. Okay, it is used for the protein manufacture. So the manufactured proteins are then sent to various places. Okay. Then when we get to the smooth endoplasmic reticulum, we want to say that the smooth endoplasmic reticulum helps in the manufacture of fat molecules. Okay, it will help in the manufacture of fat molecules. Okay, fat molecules, lipids. So it will be important for all cell function. It is important for all cell function. Okay, so it should be needed in our body. So the process, some of these proteins and lipids have been building the cell membrane. As we are not, these are present. There will be something. There are different types of things are present in the endoplasmic reticulum, like we said, proteins, lipids, okay, carbohydrates. Although those will be present in that, but in some cases it will use the cell for the production of, for the building up of the cell membrane. We will use proteins and lipids, and the process of making the cell membrane using proteins and lipids is known as membrane biogenesis. Membrane biogenesis. Membrane biogenesis. Okay. Okay, as we know, the main function of the endoplasmic reticulum is to transport of materials. So between various regions of the cytoplasm, or between the cytoplasm and the nucleus. Okay, so when we are saying about, not only it is a transport material, it is also a cytoplasmic framework providing a surface integrity, or what we say, surface for some biochemical activities of the cell. Okay, next is Golgi apparatus. Okay, I think we can run this, isn't it? Okay, you get it about the endoplasmic reticulum. So we are getting into the Golgi apparatus. Okay, so endoplasm. So ER we have already finished. Then we are getting to Golgi apparatus. This what is Golgi apparatus means. Okay, the Golgi apparatus, first described by Camillo Golgi, consists of systems of what we say membrane bound vesicles. Okay, it is combined or consists of system of membrane bound vesicles. Okay, it will be approximately parallel to each other in stacks. Okay, it will be approximately parallel to each other in stacks, and it is called as cisterns. Okay, it will contain cisterns, and stacks will be called as cisterns. Okay, cisterns will be the so these membrane of endoplasmic reticulum, and therefore it will constitute another place. Okay, it will constitute another portion. So it is known as the cellular membrane system. It will form a cellular. It will form another portion that is known as cellular membrane system. Okay, cellular membrane system. So, what we say about that? The material synthesized near the ear is packaged and dispatched to various targets inside and outside the cell through the Golgi apparatus. So, the main functions when we say about a Golgi apparatus, when we say about the Golgi apparatus, it will be storage, modification, and packaging of production in vesicles. Okay, storage, manufacture, modification. Okay, storage, modification, and packaging of products in vesicles. Okay, vesicles. Okay, so in some cases it will complex sugars may be made from simple sugars in the Golgi apparatus. So Golgi apparatus is also involved in the formation of lysosomes. Okay, you get it. So part of and also the Golgi apparatus is the portion which will help in the formation of lysosomes also. It will help in the formation of lysosomes also. Okay. So that's about the Golgi apparatus. What all we have said about the Golgi apparatus? The Golgi apparatus, it is first described by the Camillo Golgi, and so it consists of systems of membrane-bound vesicles. Okay, it consists of membrane-bound vesicles arranged approximately parallel to each other in stacks. Okay, it will call as systems. So these membranes often have connections with the membranes of endoplasmic reticulum and therefore constitute another portion of a complex cellular membrane system. So the material synthesized in the endoplasmic reticulum is packaged and dispatched to various charges inside and outside the cell through the Golgi apparatus. So its functions include storage, modification, and packaging of products in vesicles. So in some cases complex sugars may be made from simple sugars in the Golgi apparatus, and also it will help in the formation of lysosomes. Okay, so this is all about the Golgi apparatus. Okay, I think you get it. So I am going to repeat. So you don't want to write the notes because you already written it. So you are going to repeat, and we are getting into the next section. Okay, that is lysosomes. Okay. So it's also finished. Let's see lysosomes. Okay, so what we are saying about lysosomes is lysosomes are a kind of waste disposal system of the cell. Okay, this is a waste disposal system of the cell. Okay, so it is the waste disposal system of the cell. Okay, so the lysosomes helps to keep the cell clean by what we say, it will help to keep the cell clean by digesting any foreign materials. So so that's why it is known as the waste disposal system of the cell. Okay, it will keep the cell clean by digesting any foreign material as well as worn out cell organs. So what we say, foreign materials entering the cell. Okay, foreign materials entering the cell, such as what we all called about that, like bacteria or food materials that they don't needed, and already used organs, or the old organs that they have. Okay, the old organelles. So these all have already have present in the body of the cell, so it will also taken by the lysosome as a part of their function. Okay, so what will the main function? It is discussed with the waste disposal. So the lysosomes are able to do this because they contain the powerful digestive system. Okay, it have the powerful digestive system in it. So and also enzyme digestive system means the enzymes are present in the lysosomes, and the enzymes means easily it is capable of break down all organic material. So it will what we say, at the time of cellular metabolism, we can say that the cells gets erupted. Okay, cells get erupted, and lysosomes may burst, and the enzymes digest their own cell. Okay, so it is an example for the function of the lysosome. Okay, so the lysosomes may burst and the enzymes digest their own cell. So therefore, lysosomes are also known as the suicide bags of cell. Okay, only I think you are hearing only S in between that. Yes, I know that it is known as suicide bag of cell. Okay, why it's known as suicide bag of cell? It is because the lysosomes are, yes, lysosomes may burst and enzymes digest their own cell. So the lysosomes are known as suicide bags of cell. So structurally, lysosomes are membrane-bound sacs filled with digestive enzymes. So the enzymes are made by rough endoplasmic reticulum. So the enzymes are made up of rough endoplasmic reticulum. Okay. So, okay, you get it? You get it about that? So what we have said about lysosomes are a kind of waste disposal system of the cell. So lysosomes helps to keep the cells obtain. I'm just reading your textbook. So you look at in your textbook, lysosomes helps to keep the cell clean by digesting any foreign material as well as worn out cell organelles. Foreign materials entering the cell such as bacteria or food as well as old organelles end up with the lysosomes, so which break them into small pieces. So lysosomes are able to do this because they contain powerful digestive enzymes capable of breaking down all organic material. So during the disturbance in cellular metabolism, for example, when the cells gets damaged, lysosomes may burst and the enzyme digest their own cell. Therefore, lysosomes are also known as the suicide bags, that we already said. Then just reading out your textbook, so you can read out again. So get stuck by that point. Okay. And structurally, lysosomes are membrane-bound sacs filled with digestive enzymes. These enzymes are made by rough endoplasmic reticulum. Okay, I think you get it. Next, we are getting into mitochondria. Okay, 1 minute. Lysosomes we have finished. Next we are going to mitochondria. So what is mitochondria of mitochondria? Which is mitochondria is known as the powerhouse of the cell. Okay. Okay, so the mitochondria are known as the powerhouse of the cell. So the energy required for various chemical activities needed for life is released by the mitochondria. So in the form of ATP, what we know as it is ATP, it is used for the molecules. So the ATP is known as the energy currency of the cell. Okay, so the energy is formed by the mitochondria, and so it is known as the powerhouse of the cell. So ATP is formed, that means adenosine triphosphate, and ATP is known as the energy currency of the cell. As we all know that by different cycles we will attain different type of energy, but these type of energies are stored as ATP. Okay, and this ATP is used for the new chemical compounds, or will keep the new chemical compounds and for mechanical work that happens in the cell. So the mitochondria have two membrane coverings instead of just one. So what about there is an outer membrane and inner membrane for mitochondria. Okay, that is a specialty. There will be outer membrane and inner membrane for the mitochondria, and the outer membrane will be porous, Okay, it will be porous and inner membrane is deeply folded. It will be deeply folded and outer membrane is very porous. Okay, so these folds create a large surface area. Okay, so for the chemical reactions, the mitochondria is used. So the as the inner space is folded, it have large area. So the large area is used for the chemical reactions that takes place in the cell. Okay, for the production of energy. Okay, for the production of energy, that's energy. Okay, so these folds create a large surface area of ATP generating chemical reactions. Okay, so the mitochondria are strange. Okay, the mitochondria are strange organelles in the sense also that they have their own DNA and ribosomes. So the mitochondria are able to make some of their own proteins. Okay, next are plastid, and I think within plastids and vacuoles the chapter will be finished. Okay, so next is plastids. Okay, what is about plastids? Yes, so we completed mitochondria. Next is plastids. What's about the plastids? So the plastids are mainly present in not it's only present in plant cells, and there are two types of plastids, that is chromoplastids and leucoplasts. Okay, chromoplasts and leucoplasts. I think I didn't mention this thing in the class. I don't know. I forgot whether I mentioned it or not. I think said about chromoplast and leucoplast. So the chromoplast, as we know, it is colored plastids, and leucoplast means it will be colorless plastids. So what we say that the plastids which contain pigments like chlorophyll in the cell is known as the chromoplast, and which it will be used for the photosynthesis process. That's why I say that it will be only present, the plastid will be only present in the plant cell. Okay, so the plastids contain the pigment, chlorophyll are known as chloroplast. So chloroplast are important for photosynthesis in plants. Okay, and also it will contains different types of pigments like yellow or orange or pink, like that. Sorry, yellow or orange will be present within this chlorophyll or the and when we are saying leucoplast, when we are saying leucoplasts are primarily organelles. Okay, it will be primarily organelles in which materials such as the materials such as starch, oils, and protein granules are stored. Okay, so the internal organization of a plastic consists of numerous membrane layers. So it consists of numerous membranes that's embedded in material called stroma. Okay, stroma. This is known as the stroma. So the leucoplasts are primary organelles situate materials such as starch. What we say, leucoplasts are primary, as we said. So the internal organization of the plastid consists of numerous membrane layers. It consists of numerous membrane layers, and it is embedded in a material called the stroma. Okay, so the plastids are similar to mitochondria in external structure. Like the mitochondria, plastids also have their own DNA and ribosomes. Okay, so we are getting into the next section, that are vacuoles. Okay, that are vacuoles. I think I can. So what we say about plastids? Plastids are the colored substance. What we say, which contain colored or colorless things, and there will be two plastids in a plant, and it's only seen in plant. That we said, it will only see in plants. It will only see in plants, and there are two types of plastids, that are chromoplast and leucoplast. And so, the chromoplast means the colored, which will contain chlorophyll. is known as chromoplast, and which will not contain any pigment is known as, it contains proteins, oils, like that things, that is leucoplasts, and chromoplasts will help in the, yes, photosynthesis. And when we are thinking about stroma is a structure that inside the plastis, which is few layers will be there, which will be cell membranes other. So this is known as the stroma. That structure is known as the stroma. And after that, we have said this. We have said that it also it looks like mitochondria, and it contains DNA and ribosomes in the structure. Okay, so that's all we have said about the plastis, and we are getting into the vacuoles. Okay, we are getting into the vacuoles. Okay. So you just revise that what all have we studied about endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria and plastis. Okay. So we are getting into the next section, that is vacuoles. Okay. So last is completed, next is vacuoles. So about when we are saying about vacuoles, we want to say that vacuoles are storage sacs for solid or liquid condensers. For storage purposes, we are using the solid or liquid condensers, and what we say, in animal cells it will be very small. Okay, in case of animal cells it will be very small, and when we are saying about plant cells, we want to say that vacuoles will be very large. Okay, central vacuole of some plant cells may occupy what we say 50 to 90% of the cell volume. Okay, 50 to 90% of the cell volume will be central vacuole in case of plants. Okay, in case of plants. So what we say about in plant cells, where vacuoles are full of cells, but is provide turgidity and rigidity to the cell. It will give the turgidity and rigidity to the cells, and many substances of importance in the life of the plant cells are stored in vacuole. So it will contain, as we said, it will store many things in the vacuole. So it will contain amino acids, sugars, various organic acids, in some cases some proteins, like that. And when we are saying about amoeba, which is a single cell organism, as we know, the vacuole of the amoeba contains food materials. Okay, the vacuole of the amoeba contains food items, so it is known as food vacuole in case of amoeba. So and after that it will be consumed from that food vacuole. So in some unicellular organisms, specialized vacuoles also plays an important role in the exiting or the expelling of unneeded or excess water that inside the body, and from some wastes, or what we say, wastewater. Yeah, the wastewater will be expelled from the cell using these vacuoles in some unicellular organisms. So some waste or wastewater will expel from the soil. Okay, so these all are about the vacuole. So these all are the structures of the, okay, so as we studied all about fundamental types about the cell, we have studied. From this we know that the cell have importance in our life, and as we all say about the chapter's name, the name is the fundamental unit of life, isn't it? It is known as the fundamental unit of life. So it contains, or it saying about the respiration. We know that the different type of functions are going on in between inside the plant or animal body. So it will be a respiration, it will be nutrition, it will be clearing of waste materials or forming of different materials like proteins, food materials in our body. So for all this process, as we know, for all this process, it needs a cell. Okay, so cell is, from that we are concluding that cell is a fundamental unit of life, and the fundamental structure that very needed for the living organism. So it is known as a fundamental structural unit of living organisms, and also it is a basic functional unit of life. Okay, it is a basic functional unit of life, and I think you get about the fundamental unit of life chapter, and it's just a revision, and I think it's just very speedy. I think you get this clearly in the chapter section, and I just revised it to get more knowledge or more connection to that chapter. I think we will get some, what we say, some evaluation after some days, and so you should study this chapter and chapter tissue. So we will get into the new chapter on Thursday. Okay, Thursday, and we will continue the chapter. So I think you get this chapter. Thank you for listening. Stay home, stay safe.